Second type in this category will be Greek key barrel. As you see here, Greek key motifs are one half of barrels. So the way it is connected is as you see the numbers n plus 1, n plus 2, and n plus 3. The way connected could be explained this way. N beta sheet is connected to the n plus 3. But it is actually not connected to the n minus 3, not to n plus 1. It's not possible, right? Because as you see, and also n minus 1, because it must have this kind of the topology called Greek key. Okay, so right side is not possible. Left side is possible. Actually, the existing the topology. A little bit complicated, but basically two forms similar but mirror image. Actually, the present the topology is left side, not right side. This is real protein called gamma crystalline, actually being present in human eye lens. So actually composed of two domains. Okay, each domain has this kind of structure, 8 anti-parallel betas. So this is topology, okay, with the same color being used to show here. So actually they are arranged this way, but to make the situation a little bit simpler, we just from here the red one just move to this side, and the blue one at the left side actually interact with number 4 red one, but we just assume this is close to the 6. That will give you this kind of beautiful structure. Okay, two Greek key motif barrel structure. Okay, so gamma crystalline, it has two the Greek key barrel structures, two of which are almost identical. At the bottom you see the topology. Okay, really beautiful structure. But some cases does include only a single Greek key barrel, Greek key motif. In this slide, just try to compare three types: up and down barrel, Greek key barrel, and the lastly jelly roll barrel. Okay, all they share 8 basically anti-parallel betas making barrel. Okay, but the way the betas are connected are different. In up and down barrel, two neighboring betas are connected by simple loop. In Greek key, as you see, one of them just crossing the top of the column better, right? The green one also you can see 7 and 8, they just connected by the crossing loop. In jelly roll, we're gonna explain in this slide. So you see, actually, it is not belong to the two of Greek motif. You know, this loop just crossing, long crossing the loops on top and on bottom. Okay, it's much complex, but it could be understood easily when you just imagine this way. Starting with long hairpin structure with interrupted at the middle at several points. So you just try to wrap this long stretch of hairpin around column this way. Okay, starting from the end, you just put the side and then crossing the top, and then put another beta red one, and then cross the bottom with the loop region here, something like that. So in next slide, this kind of structure just giving this kind of topology. Okay, it is not belonging to the two of Greek motif. It's different, a lot complex. Okay. Actually, this kind of roll can be found in Swiss, some roll bread. That's why it's called sometimes Swiss roll motif. Hemagglutinin actually is another the cotoprotein existing on the influenza virus. You see here three protein unit trimer. Okay, so long stretched the connecting the neck, and at the end it has some the receptor head, and also at the bottom you see some transmembrane domains. Okay, actually each individual unit composed of two polypeptides called HA1 and HA2. HA1 in yellowish color in this picture has an active site at the end of this globular domain. What is the role of this receptor site on this one? It could recognize the host proteins in the step of adsorption. Okay, so it is just important in initiating infection. Okay, what about second one? HA2 in bluish color, this long stretched neck position actually is implicated in uncoating process. Okay, Coating process is critical in releasing virus itself containing genome into cytoplasm from the initial entry structure called endosome. Okay, by making disrupting the membranes, exactly the making fusion of two layers of membrane. Actually, active sites for fusing two membranes are positioned at lower positions. In total, HA2 around this position. Hemagglutinin head actually has a jelly roll structure. Okay, as you see overall, when you just consider two last beta strands as a single one, they're gonna be this way. This slide shows you the binding site of globular head of HA1 with bound inhibitor, red one, with forensic model. Okay, so there is just this is just the binding site of sialic acid located at the tip of jelly roll, the globular head shown here with sialic acid around approximately, as you see here, this portion at the center corresponding to this one. Inhibitor actually has some modified with long stretches, hydrophobic stretches in this position called R1, small one in R2. So you see this large portion actually position in the another long stretch of hydrophobic in these active regions. Okay, this is very important because it could be a ideal target for the drug design, and the study of inhibitors binding to this site have provided valuable clues and ideas for the design of molecules that are candidate drug therapy for influenza virus infections. And also, as you know, the influenza virus just every year they just produce mutant which could escape from the vaccine. It turns out antibody just binds to remove edge of this kind of protruding site. So mutant in every year they just produce mutation in these regions. Okay, but the mutant just still keep the residues in this hydrophobic binding site because this region is very important in recognizing receptor of host cells for the infection.